Thank you so much for having me here at UCL. It's my pleasure to give the statistical science seminar today. I will be presenting a computational method I'm calling infinite, which does Bayesian phylogenetic inference under the infinite sites model. And for those of you who might have studied evolutionary theory, you'll probably have heard of a lot of what this talk is going to cover before. But for those who haven't, I hope this will be an interesting introduction to a new field. The central question that phylogenetics hopes to answer is how can we use statistical methods to infer the evolutionary history of living things? We now live in a world where genomic sequencing data, in the form of DNA that can be read through lab experiments, is quite abundant. In fact, for something like the COVID-19 pandemic, we sequenced over 16 million cases and counting. And so it turns out that these DNA sequences we can collect from individuals in a population can tell us some very, very useful information about the past evolutionary histories we wish to reconstruct. And in order to apply statistics on these DNA sequences that we collect in the population, there are two key ingredients. One is a model of population dynamics, so how individuals in a population are related to each other ancestrally. And the second component is a model of genome sequence evolution. So I'll talk in the beginning of this talk a little bit about how each of those components is derived. And in particular, component one, the population dynamics model, has a lot of interesting and very rich theory behind it. Now, these models have existed for quite some time, but a very present challenge in the field is that if we wish to do Bayesian phylogenetics, which means we learn a distribution over the possible evolutionary histories and ancestral relationships between genome sequences we sample in a population, rather than a frequentist approach, which would just try to pinpoint one most likely evolutionary history, this is often very computationally expensive and doesn't scale very well to data sets with, say, thousands or tens of thousands of different sequences collected. And a big reason that that's the case is that the space of all evolutionary trees, which, as we'll see in a second, is the key object over which we wish to do inference, is a very, very large space. The number of trees with n leaves is exponential in n. So what we're going to show today is that under one very standard model of sequence evolution that applies to maybe not all, but a lot of evolutionary contexts, we can speed up these algorithms very, very significantly just by applying a number of statistical tricks, which I'll walk through towards the end of today's talk. Now, phylogenetic trees, which are our central object of study, they're what capture these evolutionary relationships, might sound like a kind of esoteric topic if you don't have a background in biology, but there are actually some phylogenetic trees out there that you all may be very familiar with. For example, this is a phylogenetic tree of the SARS-CoV-2 pandemic that shows how different variants of the virus descended from one another over time. So for example, these strains here called BA1 and BA2, they're commonly known by the Omicron variant as the Omicron variant, which was known to be more transmissible and also more harmful to humans if they got infected with it. And so these, I just want to show that these phylogenetic trees can actually tell us very useful and actionable information in the context of public health, but also in the context of human and animal evolution. So today's talk is going to capture what statistics we need to actually do probabilistic inference over objects that look like this. All right, so knowing nothing about coalescent theory or phylogenetics, let's just start from first principles and consider what would be a reasonable model of population dynamics or of ancestral relationships between different individuals in a population. Well, I'm going to lay out a very, very simple model, one that you could argue is oversimplified, but will actually do the job well enough for modeling purposes and for doing this sort of evolutionary analyses that we're interested in. So consider a population where the population size stays fixed over time. It's always just going to be 8 individuals, and where each of these columns of 8 dots, with the dots just representing individuals, represents one generation, with generations going forward in time. So for example, you could think of these 8 dots here as being our present generation, like all of us in this room. The previous column would represent our parents, the column before that would represent our grandparents, and so on and so forth. And for the sake of simplicity, we're going to imagine that each of these generations takes place in discrete time. So there's sort of a fixed interval that is the waiting time from one generation to the next. And once one generation gives birth to the next generation, we're just going to imagine that they're no longer relevant to the model. They can no longer give birth anymore. Now, of course, this is not realistic. We know that in real life, generations can overlap and all sorts of more complicated forces are actually at play, but turns out this might actually just be good enough for the sort of inference questions we're interested in. So, sort of the canonical model in population genetics, dynamics, is this thing called the Wright-Fisher model, which states the ancestor of any individual in any generation is uniformly distributed over the individuals in the previous generation. To see what this looks like, let's say we want to ask the question, who is the ancestor of this person in the most recent generation? Well, given that there are 8 individuals in the previous generation, it's this individual, the one I highlighted in medium gray with probability 1/8. It's this individual with probability 1/8. It's this individual with probability 1/8, and so on and so forth. You may be asking the question, well, wait, if you're modeling, say, a human population, doesn't every individual have 2 parents? Yes, that's absolutely true, and there are variations on Wright-Fisher processes in which individuals indeed do have 2 parents. We're going to assume, for the context of this presentation, that samples are sparse enough that we don't need to consider sort of intersecting evolutionary relationships going along both lineages that contribute to an individual. So in that sense, we can get away with considering only one lineage leading to each sampled case. Okay, so using this model, we can then say that we can then quantify the probability associated with the ancestors of all of the individuals in this most recent generation, or equivalently, say, draw from using random simulation or Monte Carlo simulation, what are the ancestral relationships leading to this latest generation. And so here's just one example of a realization drawn from the uniform distribution over who is the parent of each person in the present generation. Admittedly, this might not look terribly uniform. I drew it with mostly straight or only slightly diagonal arrows just for visual clarity, but imagine that this was sampled from the uniform distribution independently for each person in the most recent generation. Then we can just repeat this process going backwards in time, again, just drawing all of the ancestries of each individual in the second to most recent generation uniformly and independently, and so on and so forth until we there's a slide missing here, until we generate an entire tree that shows all of the ancestral relationships among people in the population, leading back to some common ancestor. So now, suppose that we observe just some subset of the individuals at present, say in the most recent generation. As was sort of indicated at this slide, the Wright-Fisher model is perfectly capable of just quantifying the probability associated with the ancestral relationships between those sampled individuals. Of course, the Wright-Fisher model will also tell us who is the ancestor of all of these unsampled individuals or people who are not in the ancestral history of the sample. But we can sort of just marginalize over all of those other ancestral relationships to compute the probability of these 5 sampled individuals leading back to some common ancestor per the tree structure that is shown on the screen here. In order to compute the probability associated with this tree, what we really want to do is compute the probability distribution of the waiting time to what are called coalescence events. And coalescent events just means the time at which any given 2 individuals share some common ancestor. It might not be the common ancestor of the entire sample, like was shown on one of the previous slides, just the common ancestor of some specific 2 samples in the population. All right, so we can do a quick probabilistic calculation to see what this waiting time should be distributed as. Looks something like this. So if we look back just one generation, the probability that the ancestor of sample A and sample B 1 generation ago is the same person would be 1/8, or 1/n in the general case of a population of size n. That can be seen by saying that, all right, let's just fix who is the ancestor of sample A. The probability that the ancestor of sample B lands on that same person is just 1 over the population size. Hence, the probability that sample A and sample B 1 generation ago do not have the same ancestor is 1 minus 1/8, or 7/8. We can then repeat this calculation moving back further generations. So the probability that those 2 ancestors of sample A and sample B, again, don't share a common ancestor moving back another generation, again, that's 7/8. You can probably see the pattern. Moving back again another generation contributes another factor of 7/8. And then finally, when they do coalesce, meaning that they do share a common ancestor, that introduces a probability of 1/8, because we need to guarantee that the ancestor of this person right here is the same as the ancestor of this person right here. As a result of this, the waiting time, moving backwards in time, for 2 individuals to coalesce follows a geometric distribution with parameter 1/n, or 1 over the population size. In this case, that would be 1/8. And now, because it's much more convenient to work in continuous time instead of dealing with these discrete generations, which would actually limit the possible space of coalescent times to some discrete set. Population geneticists would generally prefer to work with the continuous approximation to this geometric distribution, which would be the exponential distribution with parameter 1 over n. Now we can generalize this calculation to answering the question of what is the waiting time for there to be some coalescence among any number k of individuals. So in this example, k equals 5, and we're asking the question, how many generations back or how much time back do we need to go for some arbitrary pair of those 5 individuals to share a common ancestor? And the way that we calculate this is that for every pair of individuals, we have an exponential random variable with parameter 1 over n that governs the waiting time for that pair of individuals to share a common ancestor. And so given that for k individuals in the population, there are k choose 2 possible pairs of individuals that could coalesce. And so the waiting time till the next coalescence, looking backwards in time, is just the minimum of k choose 2 independent, identically distributed random variables with parameter 1 over n following the exponential distribution. And that's basically the whole thing. All that is all that you need to know to completely understand the coalescent model, which is also sometimes known as Kingman's coalescent. I'm now just going to introduce a couple of conventions that are typically used for writing and studying phylogenetic trees. First of all, phylogeneticists usually represent time going backwards, not always. The tree I actually showed earlier represented time going forwards, but for doing mathematical calculations, it's often very convenient to represent time backwards. In that way, the waiting times till the next coalescence are positive valued random variables instead of negative valued random variables. And then, sort of more to the point, it's also very common to abstract away the underlying population structure and just record the times of the coalescences as well as the structure of the tree. So that means who coalesces with who and when. This is often referred to as the tree topology. Hopefully, can I go back a slide? There we go. So yeah, hopefully you can see that even though these two diagrams look a little bit different, this diagram essentially captures all of the useful information about the previous one, which is if you look at the time of any coalescence, say like right here, of these two samples sharing a common ancestor, that's recorded as one of these branching events on the tree. And in fact, all of the different branching events, like the one here, are also encoded in the exact same way. So moving forward, we're now going to work with these abstracted trees, by which I mean just diagrams that show the sample tips together with the times of each coalescence, as well as the tree topology. So meaning the structures of these coalescences and which branches of the tree lead to which other branches. By the way, I'll just use the word branch to refer to one of these horizontal segments of the tree that just represents a chain of shared ancestors. All right, so the actual probability density that you get out of this model looks something like this. And I'm going to use notation slightly in this talk and just use a lowercase p to refer to both probability mass and probability density. I hope from context it'll be clear which one is which. All right, so let's break down this formula and how we derived it. First of all, I'll just use a lowercase x to denote the entire phylogenetic tree. That constitutes both its topology and its branch lengths. N, as before, I'll use to denote the size of the population. In the literature, this is in fact usually called the effective population size, and the reason we introduce this word effective is that it specifically refers to the number of reproducing individuals in a population as opposed to just the total number of individuals in a population, and that non-reproducing individuals will not contribute to the population dynamics process. I'm going to introduce a slight generalization moving forward, which is that we'll actually let this be a time-dependent quantity so that we can model populations that are, say, growing or shrinking over time. And that is why we end up picking up this integral in the probability density function, in that the waiting time till the next event, when it follows an exponential distribution with time varying rate, actually picks up this integral in the exponential. Then I'll let Ti denote the time of the ith coalescence. So for example, moving backwards in time, the first coalescence occurs at this point, which is somewhere between 0.1 and 0.2 units back in time. The second coalescence, or T2, occurs at this point, and so on and so forth. Then finally, this constant ci that we pick up here means the number of extant lineages choose 2. If you remember the number of lineages choose 2 factor that I introduced earlier. And this word extant lineage just means the number of branches on the tree that cross any specific slice in time. So for example, if I consider this time slice of, say, 0.1 units in the past, there would be all 5 extant lineages. At this time slice, 0.3 time units in the past, there would only be 4 extant lineages because these two have already coalesced. We're going to make one small assumption for the context of this study, which is that we're going to assume the population size, or really effective population size, is constant on each interval between coalescences. And this, you might think, is kind of a bizarre assumption. So what that is saying is that between time 0 and whenever this coalescence happens, the effective population size is some fixed quantity. Then between this coalescence and the next coalescence, we're also going to assume it's some other fixed number, and so on and so forth. And the idea here, this turns out to just be a very mathematically convenient approximation to an arbitrarily varying effective population size. It's something that was introduced in some papers in the 2010s, and we're just going to pick up where they left off in the literature. So we're going to just use the notation Ni to denote the effective population size on interval I between coalescences. Okay, so that's it for our model of the underlying population process. The second component, as I mentioned on slide 1, is a model for how genome sequences evolve. Fortunately, this is much, much simpler, and we're only going to spend a single slide on this component of the model, which is that we're going to say that mutations, or changes to the genome, arise according to a Poisson process on each branch with parameter mu. So that means that the number of mutations to occur on a branch with length Li is distributed as a Poisson random variable with parameter mu, the mutation rate, times the length of the branch, Li. How are we actually representing genome sequences, by the way? For the context of this paper, I'm going to represent them as strings of zeros and ones. As you all probably know, the actual genome contains 4 nucleotides, A, C, G, and T. But the way that we're going to abstract that to just zeros and ones is we're going to let 0 represent a non-mutated position, and 1 represent a mutated position. Now, the specific mutation model that we're going to use here, in fact, does not ever allow mutations to happen at the same site multiple times. And so we don't need to account for the possibility of a certain position on the genome having, say, both A, G, and T across different individuals. So if you want to be mathematically formal about this, we'll just represent each genome as some element of the set 0, 1 raised to the power L, where L is the length of the genome sequence. So as I mentioned, number of mutations on branch I is a Poisson random variable with rate governed by the length of that branch. And then I'm going to let this letter Y denote all of the observed genome sequences at the tips of the tree. So in that case, this would be 0, 0, 0, 0 for the completely unmutated genome sequence, which matches the sequence at the root of the tree. And then whenever we've introduced mutation at a given site, we flip that 0 to a 1, and that's how we would arrive at these genome sequences. Okay, so now I want to introduce this term of compatibility, which is to say, I'm going to use this a lot throughout the rest of the talk. And we say that a proposed tree, which I'll denote by the small letter x as before, together with the set of mutations that occur on every branch, is compatible with the observed data y if it is indeed the case that that tree would produce a given set of genome sequences y at the leaves. So for example, given that we were going to assume that the root of the tree has genome sequence 0, 0, 0, 0, we can then just read off the mutations and their sites on the tree, going from the past towards the future to determine to find deterministically exactly what the genome sequences should be at the leaves of those trees. And Y, on the other hand, is just some data set that was arbitrarily handed to you. So this compatibility thing is just checking if the proposed tree X, together with its proposed mutations M, if the genome sequences generated according to that tree match the ones that we were given in our data set. And in that way, we can write down the likelihood of our model as sort of this very trivial function that just says that the likelihood of a given data set Y is 1 if our proposed latent tree, together with its mutations, would actually realize that data set, and it's 0 otherwise. So For instance, if our data set instead had like 1111 in this entry here, well, that's clearly incompatible with this tree because there's no way that the mutations and topology of this tree could have produced a data set that looks like that. Now, I'll reiterate, the key assumption that we're going to make in this project, and this is called the infinite sites model, is that mutations always affect different positions on the genome. And this assumption ends up being quite reasonable in the case that, say, mutations are relatively rare, in which case the probability of different mutations independently affecting the same site approaches zero, or say if the genome sequence is very, very long compared to the mutation rate, again, that makes the chance of multiple mutations affecting the same site negligibly rare. And this is relevant to a lot of evolutionary contexts, including ones where maybe across, say, an entire population, this assumption is not a very good one. We can find evidence of multiple mutations affecting the same site. But if we were to restrict our analysis to some like subcluster of the population that all has a very high sequence similarity, it would become more plausible that this mutation model in fact applies. And for that reason, the fact that it's often possible to, say, filter down a data set to one in which the infinite sites model applies makes this model a very, very common one to use. Now, there is a very important structure that I want to introduce to you that sort of becomes our key for doing efficient inference under this model of the trees together with the mutation process, and that is this thing called a perfect phylogeny. So you'll notice that under the infinite sites model, these mutations essentially partition the tree into different regions that all have a common genotype. What I mean by that is, say, if we look at this red region, we can say that every part of the tree, or really every individual represented by this red portion of the tree, has the genome sequence 0000. By contrast, every individual that is represented by this small light green portion of the tree has a genome sequence 1100. And this dark green region of the tree represents the shared genotype 0011. So as a result, we can represent the ancestral relationships just between these genome sequences, independent of the topology or timing of the tree, with another graph that's called a perfect phylogeny or an augmented perfect phylogeny. The concept of an augmented perfect phylogeny does exactly that, where each node represents one genotype that's existed in the ancestral history of the sample, and each edge represents that genotype having directly evolved by way of one mutation from a different genotype. Also, each node on this augmented perfect phylogeny is labeled with the number of observed sequences, those sequences here at the leaves, representing each of those genotypes. Now, you may be wondering, why do we need to bother labeling all of these edges with one, this one here representing the number of mutations along the edge? Well, the idea of an augmented perfect phylogeny was actually something we introduced for this paper, but it's very related to a more common structure that's just called a perfect phylogeny, which is the exact same thing, except that we allow for the edges to contain multiple mutations, or equivalently, we just collapse all of the internal nodes that have exactly two edges coming out from them, and then we add the number of mutations on those neighboring edges. Another sort of more intuitive way to read this, this perfect phylogeny diagram is that we say that the light green genotype evolved from the red genotype by way of two mutations, rather than splitting it up into one mutation together with one other mutation. Now, why is this structure so important? Turns out that if we had just observed these genotypes at the leaves, but knew nothing about the tree that related those genotypes to each other ancestrally, that actually would be enough information to immediately reconstruct the perfect phylogeny or the augmented perfect phylogeny, which is kind of a remarkable fact that I unfortunately am not going to get into the proof of for this presentation. But what that is saying is that if you assume the infinite sites model, and this is not true under other evolutionary models, but only is true under the infinite sites model, we can already get this rich of information about the ancestral relationships between the different genotypes, knowing nothing about what the tree is supposed to look like. That's pretty cool. And is in fact something that we can leverage for a huge advantage in terms of doing inference over the space of these possible trees. In particular, the formalization of how this works is that, well, okay, as I said, we can construct this perfect phylogeny or augmented perfect phylogeny. They basically reflect the same amount of information from the sequences at the leaves alone. Also, if we propose some tree X together with the mutations M along the branches of that tree, that in and of itself gives rise to some other or some potentially other perfect phylogeny. And so we can say that the likelihood of the perfect phylogeny Z, which that was constructed from the data, is equal to 1 if it matches the perfect phylogeny of X together with mutations M, and 0 otherwise. So that gives us a complete Bayesian model for phylogenetic trees. We have our conditional on the effective population size, I should say. We're actually going to consider relaxing that assumption in a second, but for now, pretend that the effective population size n is just a known function. The way this Bayesian model works is that we have a prior called the Kingman's coalescent or coalescent prior over possible trees X. So it looks like this. We derived it already. Then every single branch has a model for the mutations that arise along it that follows a Poisson distribution. And then finally, our likelihood is actually this very, very simple binary function that says if the perfect phylogeny we would extract from tree X and mutations M is equal to the perfect phylogeny we can construct directly from the data, then the likelihood is 1. Otherwise, the likelihood is 0. Okay, so that's a full Bayesian model, but that's really only half of the battle. And in fact, it is this slide where our contributions actually start. Everything leading up to here, besides maybe the concept of an augmented perfect phylogeny, all of that is just very standard phylogenetics and Bayesian methods within that field. So the key question that our paper and our tool Infinite seeks to answer is how can we perform efficient Bayesian inference under this model? Because as I mentioned earlier, simply trying to navigate the space of all possible trees is brutally complex. We certainly can't do direct sampling because it's the size of that space is something like n to the n, it's exponential in n. And even by trying to do some sort of like Markov chain Monte Carlo local search algorithms, those can very often get stuck in local minima and maxima that make them pretty slow too, even though that's usually how this problem is tackled. All right, so the way that we're going to do this is we're going to apply as many statistical tricks as we can think of. So first, what we're going to do is we're going to write down a joint probability density of the mutations and the coalescences, which actually is a very elegant closed form in that we can think of it as a competing rates process. And let me walk through this formula. Oh, so I see a question in the chat. Uh-oh, the slide stopped sharing. I'm going to try resharing the screen. Can you see them now? Let me know in the chat. Yes, okay, wonderful.
Yeah, I was minimized.
Minimized.
Then you don't need to move.
Okay, cool. Okay, so step 1 is we're going to write down the joint probability density of the mutations and the coalescences as a competing rates process. I'm going to use this letter W to represent the joint object that contains both the tree, that is the topology and its branch lengths, together with the mutations along each branch of the tree. I'll let M, small m, denote the total number of mutations, and N, the total number of leaves of the tree, here 5. And this chi-I variable is just the indicator that the ith event is a coalescence. What do I mean by event? Hello? Well, since we're taking this tree to represent the joint process of coalescences and mutations, we'll take an event to be either a coalescence or a mutation. So for instance, the first event, moving backwards in time, is this mutation we have here. The second is this coalescence we have here. The third is this coalescence here, so on and so forth. So now we're going to redefine our delta TIs, those were previously our waiting times between coalescences, to be our waiting time from the I-1st to either coalescence or mutation. So for instance, our delta T3 here would be the waiting time between this coalescence and this coalescence, but our delta T4 would actually be a waiting time from a coalescence to the next mutation. Now, for some intuition as to why you actually get such a clean probability density function out of this is that, well, our waiting time to the next coalescence is governed by some rate that depends on the effective population size. Our waiting time till the next mutation is governed by some rate that is really just determined by mu, our overall mutation rate. And so as a result, and both of these follow exponential distributions. And so as we know, the minimum of a bunch of exponential distributions with different rate parameters still has a nice closed form. All right, so once we've written down that joint density for the coalescences together with the mutations, we can now apply my favorite statistical trick, which is called lumping. If you're not familiar with lumping, here's like a very simple example of what it is. So imagine you want to sample, say, the outcomes of rolling a fair six-sided die. So probability 1/6 of each of 1 through 6. Of course, in this case, we could just do direct sampling, generate random numbers that have probability 1/6 of each of these outcomes. But another approach we could use is impose equivalence relations on the different outcomes. And so suppose we say that like 1 is equivalent to 2, 3 is equivalent to 4, and 5 is equivalent to 6. What lumping would do is it would then say to sample the probability of landing in one of these equivalence classes, say like 1 or 2, compute the probability of that, and then sample the specific element within the equivalence class. So the way we do it in this example is generate a random variable that has probability 1/3 of 1 or 2, 1/3 of 3 or 4, and 1/3 of 5 or 6. Then we would choose one of those three options with equal probability, and finally, to get our ultimate samples, we'd pick the element within that equivalence class that we want to sample with its respective probability. Of course, in the instance of rolling dice, there's no reason you would ever want to do this because there's a simple closed form for the probability of each outcome. But turns out for sampling trees, it is in fact very useful. And so what we're going to do is we're going to impose an equivalence relation on trees that are identical up to the values of the delta ti's. And a good way to think about that is to imagine that we're sampling a tree that looks like this, but the tree is only defined up to these waiting times between events. So for example, we would allow this waiting time delta t3 to either shrink down or get larger. That would all be considered within the same equivalence class of trees. So how do we calculate the probability for a tree landing within this equivalence class of trees? Well, we integrate. We integrate over all the possible values of the delta ti's. What are we integrating? The probability density function for these trees. When we do this, we're actually taking an integral in, what is it, m plus n minus 1 dimensions, because there are m plus n minus 1 different values of these delta ti's, which sounds like it might be complicated. In fact, it's not. Turns out this factors perfectly in that the integral of the product just becomes the product of a bunch of integrals, and it simplifies nicely. It's just an integral of e to the minus something from 0 to infinity. Okay, so we get a nice tractable probability density for these equivalence classes of trees that are the same, except for possibly the values of their delta ti's, their waiting times. Turns out, you can see from this calculation, there's actually many other ways to see this as well, that mu, the mutation rate, and the effective population size n are not identifiable from one another. The reason this is true is that we can write the entire probability distribution in terms of the product, mu times the effective population size n. So as a result, moving forward with completely without loss of generality, we will just assume that mu equals 1. All right, and our final step 3 is that we're going to do more lumping. And the way that we're going to do this is we're going to notice that we can also lump together trees based on which coalescence happens within a genotype. And so the idea behind this is that, well, we know the probability of, say, the tree shown on the left here is the same as the probability of the same tree, except that imagine that 2 and 3 had coalesced first, and then they coalesced with number 4. And once we do this, we can actually represent a tree just in terms of the sequence of mutations and coalescences that happen on that tree, together with which genotype they affect. So for example, in this tree, the first event is a mutation affecting the light green genotype, turning it into blue. The second one is a coalescence occurring in the dark green genotype, followed by another coalescence in the dark green genotype, and so on and so forth. Turns out that this lumping still admits a tractable probability mass function, and in fact, the only thing that we have to multiply the previous probability mass by is this additional combinatorial factor, that is the number of current extant lineages of a given genotype, choose 2, for each coalescent event. Now, I'm going to use this letter U to refer to this thing called an evolutionary path, which technically speaking is a subset, or really an equivalence class, of the previous object that we introduced, V, that are the same up to the choice of pair of lineages involved in each coalescence. That's a mouthful, but a better way to think about it is an evolutionary path is simply one of these sequences of genotypes in which each evolutionary event occurs, that being a coalescence or a mutation, together with the label of that as either coalescence or mutation. And that is the space over which we wish to conduct our Bayesian inference. Now, sort of the punchline here is that while conducting inference over the space of like trees that look like this is very hard, as we stated, that was like a combinatorially large space, conducting inference over these sequences of mutations and coalescences is in fact very, very easy. It's so easy that we can explore this space with a single Markov chain Monte Carlo move. In case you're not familiar with MCMC, this is an algorithm for doing Bayesian inference over a given space that involves perturbing the state such that you generate a Markov chain whose stationary distribution is equal to the posterior distribution that you wish to sample. Very often, these Markov chain Monte Carlo algorithms employ lots of different and very complicated moves that sort of are chosen heuristically such that the Markov chain has a better chance of converging to the correct posterior in a reasonable amount of runtime. But we were able to do this with just one single move, where you choose at random two indices of this sequence, indices here going from 1 through 8, representing all of the coalescences and mutations, and you just propose a swap to those indices, then accept or reject them with the Metropolis-Hastings criterion, the standard criterion used for MCMC. That turns out to be good enough in this context to very rapidly converge to the true posterior distribution over trees. So this is where we've significantly deviated from the norm in Bayesian phylogenetic inference in that existing methods such as Beast, which is often considered the state of the art, as well as even more modern methods that are in fact a lot faster already than Beast, like Delphi, which was introduced by my old lab in a paper last year, they do explicitly manipulate these full trees. So those manipulations might look like taking one branch, plucking it off, and trying to reattach it somewhere else. There are other moves that might adjust, say, the time of a coalescence or something like that. They're sort of chosen to align maybe with our intuition for what would be a reasonable tree rearrangement, but at the end of the day, really are just heuristics. We don't need any of that in this method. Finally, I want to state that even though the sort of core of the infinite algorithm is explicitly sampling these sequences of coalescences and mutations, there's a trivial Monte Carlo sampling step that you can do at the end to take one of these sequences of coalescences and mutations and randomly convert it back to one of these full timed trees, often called Kingman trees. So don't worry, even though it looks like we're sampling something a lot more abstract, you can always apply this transformation at the end to get back the posterior distribution over the trees. All right, so this has been a lot of theory, but does this work? Well, as far as we're aware, yes, it does. First, what we did is we simulated some trees with constant effective population size, so not time varying, and in fact didn't do any inference over the effective population size, and checked whether or not the distributions recovered by our model, shown in blue, agreed with the distributions that were obtained from the existing software Beast, which are shown in brown. And up to some statistical noise, yes, we indeed saw that these distributions agree. And the sampling efficiency, which we measured in effective sample size per second of wall clock time for our method, was significantly higher than Beast. To give you some better context on how much faster, fortunately, we saw our efficiency improvements, in fact, scale with the total size of the tree measured in the number of leaves. So, for example, simulating trees with only 32 leaves or 32 taxa, we only saw a speed up of about like 1.3 with over Beast 2, but as we scaled to larger and larger trees, say with 512 leaves, our method was able to perform up to 226 times faster than the current method Beast 2, which is widely considered the state of the art. I will, of course, note that the key difference between what we're doing in Beast 2 is that we assume the infinite sites model, Beast 2 does not. So Beast 2 certainly is a more flexible tool in that sense. But for context in which the infinite sites model applies, this is, I hope that this convinces you, I should say, that infinite is a good option. Now, I haven't discussed too much about inferring the effective population size. We kind of just assumed that it was a fixed function n throughout this presentation, but it turns out we can also do Bayesian inference over that. And here we saw even more significant increases in effective sample size per second as compared to Beast 2 under certain models. And moreover, we were able to cover these kind of rich trends in effective population size, such as correctly inferring that an effective population size either decays or stays constant or grows over time. Again, matching the results of Beast. Let's see one thing in the chat. I'm a little confused. You said MCMC only has one move, swapping events, and we are only inferring the order of the events. But you just showed the posterior over coalescence times. When did we infer that if we were only looking at the order of events? Yeah, that's a great question. So the I guess what I should say is that the core of the algorithm here is doing MCMC just over the orders of the events. Once we obtain our posterior distribution over the orders and genotypes of those events, we then apply this direct Monte Carlo sampling step to convert that back into a full tree, where this tree contains the complete topology, as well as the times of each event. So it's sort of like an extra Monte Carlo sampling step at the end that is negligible in runtime compared to the MCMC step. Yeah, and then finally let's see. So two-step MCMC, sort of. So the only MCMC that's done, yeah, so it's MCMC on the order, and then I would say just MC, not MCMC on the times, in that we don't actually need to construct a markup chain to obtain back the times. It's just directly sampling from very simple distributions. So, for example, those last sampling steps are just drawing random variables from either an exponential distribution or a discrete uniform distribution. So we don't even need MCMC to do that. Um, Okay, cool. So then finally, we applied this to some real data. This was actually my first time studying human evolutionary data. These are effective population size trajectories for various human groups in the past based on the mitochondrial DNA record. My background, funnily enough, is in viruses, but it's kind of cool to see how the same methods can apply both for viruses and humans, as well as plenty of other organisms. And the reason we decided to do this is that often in viruses, we have samples that are collected at very different points in time. And there is a way we could generalize this method to that context, but it's just not something that was in scope for this paper. That being said, for human populations, it's perfectly fine to assume that all samples were collected at the same time, because that's any spread in those sample times is negligible compared to the total evolutionary time or the time of the most recent common ancestor. And what we were able to do is pretty accurately recapitulate the effective population size trajectories as inferred by Beast. And you may be wondering, why are we seeing some discrepancy here? Well, it's because of that infinite sites model assumption, which is that in order to run infinite, we first had to take the genome sequences from the humans and restrict them to sites on the genome that are compatible with the infinite sites model. And so that masking loses a little bit of information that does cause some divergence in these evolutionary histories. That being said, it's not a lot of information, and these simulation studies show that, which is we simulated some data sets that had different levels of what are called homoplasic mutations, which are just mutations that affect the same site on the genome and are hence not compatible with the infinite sites model. And what we found is that even with growing numbers of homoplasic mutations, in this case, about a third of the data set consisting of homoplasic mutations, even though our curves and confidence intervals for the effective population size, as well as other posterior estimates, did look a little bit different between these examples, they still are good at recovering general trends in effective population size. And in fact, consistently, the 95% credible intervals for these parameters contained the true value. All right, so what are the takeaways? We can make Bayesian phylogenetics, assuming the infinite sites model, very efficient by sampling this coarser structure that we're calling evolutionary paths, run MCMC on that, then by this sort of post hoc Monte Carlo sampling step, convert those evolutionary paths back into complete phylogenetic trees. Moving forward, we're interested in some generalizations of this approach. One pretty straightforward one would be some more sophisticated models of the effective population size. You might have remembered that we're assuming the effective population size to be constant on intercoalescent intervals. We can definitely relax that if we would like to. It just requires a bit more work. In addition, as I mentioned, to generalize this to, say, viruses, it would be important to model samples being collected at different points in time throughout history. Then one thing that we actually looked at for quite some time in this project and then discarded after trying for about 6 months was different inference methods beyond MCMC. And in particular, we were interested in trying out sequential Monte Carlo as our inference method, but turns out that this just resulted in this thing called particle degeneracy, where you see that all of your samples actually end up converging to what looks like just one sample, which makes it pretty much useless. But we certainly didn't exhaust all of the possible ways of implementing sequential Monte Carlo, so I'd be interested to come back to that. And then finally, on the theoretical side, we're interested in considering a generalization to finite sites evolutionary models. I think this is very non-trivial because you lose the ability to use that nice trick of the perfect phylogeny, where you can construct the perfect phylogeny both from the data as well as the inferred tree. That being said, you might still be able to retain the idea of representing trees just in terms of their sequences of coalescences and mutations and which genotypes they occur in. Then finally, it would be interesting to do a more applied study on which data sets are compatible or almost compatible with the infinite sites model. My suspicion, just from having worked in the field for some time, is that infectious disease outbreaks, particularly viral outbreaks, might be a very good candidate because often they span relatively short time frames and so don't have the chance to pick up that many mutations, and hence the probability of them picking up homoplasic mutations is quite low. But it would be great to actually quantify the extent to which the infinite sites model applies to those outbreaks. Finally, just want to thank various folks who read over our manuscript, including my former PI, Pardeep Sabeti from Harvard, and finally, could not have done this without my primary advisor at Stanford University, Julio Palacios, who guided this project.